Hello and welcome to BioRules video lessons. Previous class we took a look on prokaryotic cells. Today we are going to see eukaryotic cells. Eukaryotes are the animals, the plants, the fungi, the protists, which include the protozoa like an amoeba and algae. And what these cells have in common is that they have a true nucleus. They have an envelope that surrounds the nuclear material. They have the DNA surrounded by a membrane. They also have membrane surrounding, forming many organelles inside. So they have membrane-bound organelles like mitochondria, like chloroplast, Golgi, and others. So they have some. In the end, what they are going to be having is that they have lots and lots and lots of membranes inside. Okay. Here are a few examples of eukaryotic cells. These are animal cells. We have the red blood cells in red. We have the white blood cells, which are these ones here and these ones over there. Those are the white blood cells, the cells from our blood. Okay, a typical animal cell will look like this. It will have a membrane. It will have a nuclear envelope that is double membraned, have two membranes and pores, have openings to the cytoplasm. It will have chromatin, which is the DNA inside with associated proteins, and they also have a nucleolus, a condensated area over there. Surrounding the nucleus, you're going to be having rough endoplasmatic reticulum. It's a big series of bags, and these ones are going to be called rough because they have ribosomes in it, to make it look like no rough. We also have smooth endoplasmatic reticulum, so it's bags surrounding the nucleus, continuous to the nuclear envelope, but without any ribosomes. Okay, getting material vesicles from those nucleus, you're going to have the Golgi apparatus, those structure that looks like this. They are very characteristic when we look at them. Okay, they are going to be having mitochondria, which are these bacteria-like organelles that have a specific shape that we're going to study with more details later. Okay, it's double membrane as well. You have an outside one and an internal folded membrane. We have, in animal cells, we're going to have centrioles, these tiny little cylinders that are very important for, among other stuff, for cell division. Okay, and we have all over the cell, not only in the corner, I just draw here in the corner to make it easier, ribosomes. So these cells are going to be containing free ribosomes in addition to the ribosomes that are there in the endoplasmatic reticulum. So let's give name for things. Cell membrane is that. So you already saw this is going to be the cytoplasm. Okay, this one is the nuclear envelope. So it's the membranes double surrounding the nucleus. We have an opening here, which is the nuclear pore, which now connects the cytoplasm to the inside of the cell. We have there chromatin. Chromatin is DNA associated with proteins in eukaryotes. Eukaryotes, the DNA is not naked, so it's inside of a nucleus, and the DNA is mixed up with proteins. We have there a nucleolus. It's an important part for the manufacturing of some important molecules like transport RNA. More details about that later. Okay. Here we have the RER, rough endoplasmatic reticulum, with its associated ribosomes. There, the centrioles that I have showed before. Now we have the drawing there. The centrioles that are going to be important, among other things, to make microtubules for cell division. There we have the SER, the smooth endoplasmatic reticulum, which is an endoplasmatic reticulum, in the same way as the rough, continuous to the cell, to the nuclear envelope, but in this case without ribosomes. There are the beautiful bacteria-like mitochondria that are going to be very important for every single cell to be able to deal with energy. Here we have the Golgi apparatus, which ultimately is a series of bag discs that come from vesicles from the endoplasmatic reticulums, and they are going to be doing stuff over there, they compacting stuff, producing, and they are going to be sending bags outside, which are going to be called vesicles. So there we have the Golgi, and here we have a vesicle from the Golgi that's going to be exporting material. Okay, there we have a free ribosome, and in this case here, as I told you before, these ribosomes are bigger than in prokaryotes. They are ATS. S is a unit that refers to size, but no, actually it's a unit of weight. Okay, so this here is a typical animal cell. So to talk about the functions, let's take a look here. Function of the cytoplasm is the same as before. No, it supports the organelles and you have the chemical reactions happening in there. Okay, the membrane is the same as in prokaryotes, controls the in and out of the cell. The nuclear envelopes, it contains the chromatin, contains the DNA and the proteins associated with the DNA. Nuclear pores is the in and out of the nucleus. Okay, nucleolus is a tRNA and ribosomes factory, so it's where the cells form ribosomes and tRNA. The chromatin itself is DNA plus proteins, more details on that later. Centrioles, they are microtubes formation centers. They are places where the cells make microtubules. Okay. Mitochondria, very important organelles for aerobic cell respiration. No? So most cells, eukaryotic cells, they are going to be having mitochondria, including plants. Okay, they will have mitochondria as well. Ribosomes, again, in the same way as in the prokaryotes, they are for protein formation. The free ribosomes are for the cell use. So the free ribosomes make proteins that the cells are going to be using themselves. And the rough endoplasmic reticulum with its ribosomes, it's also for protein production, but they produce proteins that are for export. They produce proteins that are to be sent out of the cell. The smooth endoplasmic reticulum is also related to production, but not production of proteins because they don't have ribosomes. So what do they produce? The other two macromolecules. They are going to be producing carbohydrates and lipids, so basically sugars and fats. Also, again, mostly for export. Okay, Golgi apparatus. They get material from rough endoplasmic reticulum, smooth endoplasmic reticulum. They put them together, they assemble them, they pack them, they get it ready to send it for export. So basically, they are protein, lipids, carbs, package center, and they export to the vesicles. Vesicles are export material, usually from Golgi, but you can have vesicles from other places as well. But these vesicles shown in here, they are for exporting material that come from Golgi. This is the function, these are the functions of parts of the eukaryotic cell, the animal eukaryotic cell. So here you have a few more images for you to see how they look like in 3 dimensions. You have this smooth endoplasmic reticulum, these bags without the ribosomes. We have the rough endoplasmic reticulum, these bags with the ribosomes. We have the Golgi here, which basically looks like flattened sacs with vesicles that come from the endoplasmic reticulum and vesicles that go away. Have the mitochondria there, the centrioles, they're all here beautifully and nicely. Okay, another image for you to be able to see the structures that you have inside. Okay, this here is a transmission electron micrograph showing some structures. So we have here the Golgi, not so visible in this image, but there are others that you can see. Here we have centrioles. This darkish stuff here, they are the mitochondrion. We have endoplasmic reticulum, not very clear in this image. And of course, the star of the show here, we have the nucleus. Usually the nucleus is round. Okay. Here, another image for you to be able to see, and I bet you can see here the nucleus. I bet you can see here mitochondrion, here in this place, this gray stuff. These black dots are vesicles. This cell probably produces lots of material to export. Here we have rough endoplasmic reticulum, lots of bags going and coming back here. This is the rough endoplasmic reticulum. So this is probably a cell that's producing lots of protein by the amount of endoplasmic reticulum, by the amount of rough endoplasmic reticulum that it has. Okay, additionally, let's take a look on the plant cell structure. They are very similar. They have a cell membrane, but they are going to be having differently from the animal cell, they have a cell wall. Okay? Inside, they're going to be having all the rest is going to be practically the same. They have nucleus, they have rough endoplasmatic reticulum, they have smooth endoplasmatic reticulum, they have Golgi, they do have mitochondria. Be careful, this is a common mistake that students think that because plants do photosynthesis, they don't have mitochondria, but they do have mitochondria. They do aerobic cell respiration. But in addition, plant cells will have these green structures called chloroplasts, which are photosynthesizing organelles. They are the organelles that will do photosynthesis. They transform light into organic molecules. And many plant cells might have a big central vacuole, which is nothing more than a very big vesicle. Okay, so this is the structure of the plant cells. So the names are here: cell membrane, the cytoplasm over there, the nuclear envelope, the nuclear pores, same thing as before, the chromatin, the nucleolus, the rough endoplasmatic reticulum, smooth endoplasmatic reticulum, the mitochondrion, all the same. What's new? Oh, sorry, Golgi also all the same. Vesicles all the same. What's new here is the chloroplast. What's new here is the large central vacuole. And what's new here is a cell wall. Okay, by the way, the cell wall in plant cells, they are made of cellulose. So they are different from prokaryote cells wall that are glycoproteic. The plant cells wall, they are glyco. So they are made of only cellulose. They are made of a sugar, a special sugar called cellulose. The same that we make paper. Okay? Of course, they also have ribosomes. I was forgetting to put them here. So, 3 ATS ribosomes. They are eukaryotes, so they have ATS ribosomes as animals, as fungi, as any eukaryotes have ATS ribosomes for the internal production of materials for the cell. Taking a look on the functions, so everything is here drawn. I'm not going to be repeating myself with the organelles that you already saw in animals, but I'll show you the function of the cell wall is to keep the shape and avoid this from letting the cell to burst, to explode with too much pressure. Okay. The chloroplast I already mentioned, the function is to do photosynthesis, and the large central vacuole is for storage. One thing that you might have realized is that these cells here are missing the centrioles. Plant cells don't have centrioles. So they will do cell division producing microtubes, but they do that without an organizing center called centrioles. Okay? One image here for you to see the structures beautifully and nicely, and here some microscope images for you. Here on the left side we can see very clearly the cell wall and many chloroplasts. Here the same. We see in this scan electromicrograph we can see some cell walls, and we see some balls as well that are probably the chloroplasts as well. Okay. One thing that you should be able to do is to compare cells. So first thing, compare plants and animal cells. How are you going to be doing that? First, what about nucleus? Think about it, and yes, both of them are going to be having. What about membranes? Think about it, and yes, both of them will have membranes. What about rough endoplasmatic reticulum and smooth endoplasmatic reticulum? Yes, they are both going to be having it. Golgi? Yes, both are going to be having it. What about mitochondria? Do plants have mitochondria? Do animals have mitochondria? And yes, both of them have mitochondria. Okay, centrioles? Plants don't have centrioles. Animals have centrioles. Okay, cell wall. Plants have cell wall, cellulosic cell wall, and animals don't have cell walls. They keep their shape by packing cells together. They avoid them from bursting using other strategies. What about chloroplasts? Yes, only plants have chloroplasts, animals don't have chloroplasts. What about vacuole? Plants have large central vacuoles, and animals don't have large central vacuoles. So you need to be able to compare plants and animals, and remember, comparing means telling the differences but also telling the similarities between the two things that you're going to be comparing. Another comparison that you should be able to do is to compare prokaryotic cells with eukaryotic cells. So let's take a look on one by one. What about membranes? Think a bit. And yes, both of them you have membranes. What about cytoplasm? Yes, both of them have cytoplasm. What about membrane organelles? And the answer here is prokaryotes don't have membrane organelles, apart from some cells that have a mesosome, which is an enfolding of the cell membrane, which could be considered a membrane organelle. It's not separate from the cell membrane. So yeah, in the end, no, prokaryotes don't have membrane organelles. Eukaryotes have lots of membrane organelles. What about a nucleus? The same. A nucleus is made of membranes, so prokaryotes don't have membrane organelles, they don't have a nucleus. Eukaryotes have nucleus. Plasmids. Prokaryotes have plasmids, eukaryotes don't have plasmids. Eukaryotes are going to have chromosomes, tapes of DNA inside of the nucleus. They don't have little rings of DNA in their cytoplasm. Ribosomes, do they have ribosomes? If you thought yes, you're right, but the point is, are they equal or are they different? Prokaryotes have 70S ribosomes, eukaryotes have larger 80S ribosomes. Okay? Pili, only prokaryotes have pili, eukaryotes don't have this pili projections going through the cell wall, in the plants or animals, so they are going to have different things. Flagellum, this is something that students sometimes get confused. I don't know why. It's very common for students to say that prokaryotes have flagellum. Yes, and they're right. They have simple flagellum. But what you have to remember is that eukaryotes also might have flagellum. They are going to be having a more complex flagellum that are going to be microtubuled. For example, spermatozoa and enterozoids, they have flagellums. So remember, both of them will have flagellum. The difference is that the flagellum in prokaryotes are simple and the flagellums in eukaryotes are microtubuled. Okay? Cell wall. So, prokaryotes have glycoprotein cell wall, very good, but eukaryotes might have cell wall. Plants have cellulose cell wall, fungus have chitin cell wall, and animals don't have cell wall. So that's important, mention that, okay? What about size? Prokaryotes are tiny, very small. No, they are usually 10 micrometers or even smaller. And eukaryotes, they can be much bigger. There are some small eukaryotic cells, but there are exceptions. Most eukaryotes are going to be bigger, 50 to 100 or even more micrometer size. And the reason for being bigger is that eukaryotes have much more membrane. Not membrane outside, but they have membrane also inside. So they can make more process happen in faster ways if they play with membranes in a different way than compared to the prokaryotes. So eukaryote cells, they can grow bigger in part because they have lots and lots and lots of membranes inside. Okay. If you want to see a video, I suggest you see this. We'll see in class as well, but there's a very good short video that shows the inner life of a cell. Take a look on it, try to identify organelles, because later in class we are going to play this video and you're going to tell me which organelles you have been able to identify. I hope you had fun and I'll see you in class or I see you in another video. Bye bye.